Good morning, everybody, and thank you for joining us. I want to introduce myself first. I'm David Fulton. I'm the Assistant Unit Leader in the Minnesota Cooperative Fish and Wildlife Research Unit. I'm a USGS U.S. Geological Survey research ecologist, but really what I do is focus on conservation social science. And I'll be presenting a kind of a high-level overview of a proposed project that we're hoping to get started in January. And the focus of this project is going to be understanding attitudes towards RNA interference methods for controlling invasive carp, more recent invasive carp, not common carp, and also zebra mussel. I do want to take the time to acknowledge partners on this project that are involved and partners that invited me into the idea of this project. And these are all U.S. Geological Survey colleagues at different ecological and science centers. So we have John Amberg at the Upper Mississippi Environmental Science Center, along with Christopher Merkes there at the same location. Also Kathy Richter down at the Columbia Environmental Science Center with USGS. And finally, Nicholas Cole out at the Fort Collins Science Center. Myself and these partners will all be participating in this project with in-kind contributions from U.S. Geological Survey to support our efforts on this research project. In addition, this research project would support a 2-year research associate who will be a University of Minnesota employee. The background for this project is that I think most of us know that new synthetic biology, genetic engineering technologies such as RNAi have been developed, and especially in the last few years, rapid development of these technologies with a lot of promise to control invasive species. Because of this, there's growing calls from researchers who are exploring these techniques, as well as conservationists, to explore the benefits and use of genetic biocontrol technologies, including RNAi, especially when it's focused on control of invasive species. In order to move forward with the development and potential implementation, we need to understand first professional and stakeholder perceptions about the benefits and risk of using RNAi for genetic biocontrol. This understanding will help guide policy concerning its potential implementation into the future. This study is focused squarely on that need, and the objectives of the study are to investigate stakeholder awareness of RNAi techniques, identify concerns that might be out there among the stakeholders and professionals about RNAi, including what information they would want to inform their decision-making concerning whether to support the technology, and how they would like to see the technology potentially used in controlling invasive carp or zebra mussels. In addition, we want to understand and to describe the attitudes towards the perceived downstream risk of conducting RNAi control of invasive species, and identify the potential harms from this technology that stakeholders would want to make sure are rigorously studied. So these objectives, again, are focused on trying to understand upstream concerns, primarily from key stakeholders and the professionals that are studying the use of RNAi techniques. The methods that we would use are much more in the vein of qualitative social science methods and techniques that are really useful for exploratory studies. We'll conduct in-depth, semi-structured interviews and focus groups with professionals and stakeholder groups to develop a grounded theoretical understanding and conceptual map of the issues from their perspectives. This would involve engagement of up to about 100 different individuals in the upper Midwest and nationwide to understand what some of these key concerns are, again, among the scientists that are developing the techniques, as well as key stakeholder groups. We'll transcribe interviews and workshops that will then be thematically coded to identify representative themes under each of the goal topics, using both expected and emergent coding practices. From this, we hope to develop a conceptual map or model of the key themes surrounding this issue of use of RNAi and how these key themes are related to one another. Along with the methods, identifying our important part of these methods are identifying who the participants in the study would likely be. We see as the potential stakeholders, scientists in academia, government, and industry studying invasive carp, as well as zebra mussel. Also, scientists in these same institutions that are studying RNAi for control of invasive species, particularly newly invasive carp and zebra mussel. We will focus on state, federal, and tribal managers that are working on invasive species issues and are concerned with invasive species. We'll also have representatives of conservation, our environmental non-governmental organizations, as well as the broader public. The outcomes of this study, what we're hoping to achieve is to inform future efforts to develop best practices for implementation of RNAi and provide key information needed to appropriately engage stakeholders to foster support. I would include with this engagement is a more detailed, specific studies of where the general public might be. But we perceive that a first key step is really getting this broader understanding from the professional community and the stakeholder community of what perceptions of risk and benefits and other concerns might be with the use of this technology. Again, a very high level overview of this project, but more than happy to answer any questions that folks might have. Yeah, and just to tie on, we are just finishing up another study where we did drill into the public and key stakeholders, more specific attitudes towards the general use of genetic biocontrol. But in that study, we were not really able to get into specific techniques like RNAi because of the complexity of the techniques, or CRISPR, or some of these other techniques that might be out there. So this will be a first of its kind study where we really are drilling into RNAi, trying to better understand what some of these concerns and promises of the technologies might be from the researchers themselves and from the managers that would be ultimately involved with implementation of any such control techniques.
So then what is the value of this in-depth qualitative approach used for this study?
These are very new technologies, literally just being developed in the last few years. It's still uncertain what implementation would look like. The other thing is that we haven't had those deeper conversations in the professional community about what potential unknown risk might be. And so what this will do is a professional community help us understand what concerns might be out there. That's going to be essential as we move towards any implementation of using these technologies. State and federal agencies will be looking for guidance about what the policy implementation will look like. So we see this as a first step of providing upstream information about moving towards any policy framework around the use of techniques such as RNAi. And one thing that I would say, if any folks have additional questions, feel free to follow up. You can contact me via my email at dcfulton@umn.edu. but happy to answer any questions as we get going on the project. Again, preliminary contact with potential stakeholders and other partners this fall. And the idea is to get going in earnest in the spring with the placement of a research associate.
So just a reminder to everyone in the web room too, this is a lightning round. And so we have 3 presentations today. We are moving to our second, presented by Gretchen Hansen on zebra mussel impacts on fish mercury concentrations.
Thank you, everybody for being here, and thanks to MESARC for hosting this and giving me the opportunity to talk about a new project. So my name is Gretchen Hansen. I'm an assistant professor in the Fisheries, Wildlife, and Conservation Biology Department here at University of Minnesota. I'm going to talk to you today about a new project that we're starting up in January, looking at zebra mussel impacts on fish mercury concentrations. And as you can see from the title slide, this is a collaborative project with a number of partners. We have a few other researchers here at the University of Minnesota, Paul Radomski and Heidi Rantala from the Minnesota DNR, Jenny Brentrup from the Minnesota PCA, Karen Ackley, Mark Luring, and Adam Ray from the Great Lakes Indian Fish and Wildlife Commission, and Ty Wagner from USGS Co-op Unit at Penn State, and Aaron Schlipp from North Carolina State University. So lots of partners here in Minnesota and some external expertise to help us with some of the statistical modeling. All right, let's see if this is working. Okay, good. All right, so for this crowd, I certainly do not need to tell you that what zebra mussels are, I don't think. Zebra mussels are prolific and highly impactful invasive species here in Minnesota and throughout North America. They can form, sometimes, not always, but they can form really dense colonies, dense mats on the lake bottom, and have really big impacts on water clarity by sucking those nutrients out of the water column and kind of depositing additional nutrients on the lake bottom. And those ecosystem level effects of changing the nutrient flow in lakes has a lot of implications for the whole lake ecosystem, including higher trophic levels such as fish, which is an area that I work on a lot, me and the students and researchers in my lab. And those impacts on higher trophic levels are somewhat less understood and a little bit more variable than what we know about, say, zebra mussel effects on water clarity, but we're starting to learn more about that. And before I tell you more about impacts on fish, I just want to take a moment to point out one of the reasons why I'm really interested in understanding zebra mussel impacts on fish, and particularly walleye. I do a lot of work on walleye. Is that, of course, walleye are important in Minnesota, they're the state fish, but also zebra mussel invasions in Minnesota are much more likely to happen in lakes that contain and are managed for walleye by the Minnesota DNR than lakes that are not managed for walleye. So there are just about 300 lakes with confirmed zebra mussel infestations in the state, and 77% of those are lakes that are actively managed for walleye. So that's quite a lot. And so the infestation rate of zebra mussels in walleye lakes is quite a bit higher than in lakes that aren't managed for walleye. And those non-walleye lakes are actually the vast majority of lakes in the state, but the walleye lakes are much more likely to be invaded. And that makes sense because these lakes tend to be bigger, just generally really popular destinations for boaters and anglers. And so it makes sense. And also it's sort of an impetus to really understand the impacts of zebra mussels on walleye and other fish that are targeted by anglers throughout the state. So because this is a lightning talk, we don't have time for me to tell you all the different ways that zebra mussels impact fish populations and walleye. I will say that understanding these impacts has been a big focus of my lab, and through MESARC funded research and other projects, we've really started to understand more about how zebra mussels impact fisheries in Minnesota and beyond. And so happy to talk about those at any time with anybody. But for today, just know that we've documented links between the ecosystem impacts of zebra mussels, effects on fish populations, and then how those effects kind of show up in the fishery in terms of like what anglers see in their walleye populations and in the fish that they're trying to catch. What we're going to talk about today is a relatively new impact that researchers in my group and in other places at USGS have been interested in exploring, and that is the potential for zebra mussels to increase mercury concentrations in the tissues of fish. And so I'm going to give you a little background and explain why we think this might be possible, and then tell you how we're going to look at it through this project. So just quickly, you probably know mercury is a human health concern. It is a neurotoxin, and it is particularly a potential health issue for children and women of childbearing age who might become pregnant. And one of the major sources of mercury for humans is through eating fish, particularly top predator fish, where mercury bioaccumulates in their tissues. And so again, if you've ever looked into this or gone fishing in certain places, you might see that there are advisories or signs providing guidance about which species and how many certain populations should eat or should limit their consumption of in order to reduce the risk of those health impacts. Okay, so what does that have to do with zebra mussels? There's a couple lines of evidence that led us to think, I wonder if zebra mussels could be affecting fish tissue mercury in Minnesota lakes. So number one, there's been documentation in Minnesota and in other states in the Great Lakes region of recent increases in mercury in the tissues of some fish in some lakes, following really widespread reductions in mercury concentrations due to changes in atmospheric deposition. So as we, as a nation and as a global community kind of understood where that mercury was coming from certain industrial processes and being emitted into the atmosphere, we've gotten a handle on that, really reduced the emissions from those industrial sources. And we saw over time a decline in mercury in fish tissues from the 70s through kind of the mid 90s, early 2000s. But then since the 1990s, mercury concentrations have been going up again in walleye and in other species in Minnesota and throughout the Great Lakes region. And so there's a lot of variability in those trends. There's a lot of hypotheses about what might be going on, a lot of factors at play, and I'll talk in a minute about what some of those might be. But potentially, zebra mussels might be playing a role. We also have evidence from the Great Lakes that top predator fish there experienced increased mercury concentrations following invasion of zebra mussels and quagga mussels. And the researchers in that case really documented the mechanism that was going on there that I'm going to explain to you in a second. And so work in my lab has been starting to investigate this over the past couple of years. We have some preliminary work showing that there is elevated mercury in fish from a small number of lakes containing zebra mussels. And so this project is designed to really dig into that and see how widespread that effect might be and look at variability among lakes in how zebra mussels impact mercury. So what controls fish tissue mercury? I am not a biogeochemist, and I am assuming most of you aren't either, so we're not going to go into the whole mercury cycle, but I will just say the controls on how much mercury ends up in fish that you might be interested in eating are there's a lot of things that control that. So there's atmospheric deposition, how much is coming in from the air. There's also an influence of how much mercury is in the watershed already, kind of in the soils, and how that mercury in the soils is being input or run off into lakes. Once mercury is in a lake, there's differences in how available it is to fish populations that are related to water chemistry, water clarity, dissolved oxygen, as well as food web pathways and how fast the fish are growing. And we know that zebra mussels influence a lot of these things that can influence how much mercury is in the water column and how accessible it is to fish. So briefly, zebra mussels, when they establish, if they form these dense beds on the sediments, they can reduce the oxygen in those sediments. And in doing so, increase microbial activity that releases mercury in its available form to fish. At the same time, fish are eating more in those nearshore zones as their prey is reduced in the offshore zones. So through those two pathways, they can increase the availability of mercury that might already be in the lake tied up in the sediments to fish that we might be concerned about eating. So, okay, our objectives for this project are to, given that background, quantify the effect of zebra mussels on fish mercury concentrations in Minnesota lakes, try to develop a predictive model of mercury concentrations in the lakes and how it relates to zebra mussel invasion, and then collaborate with our partners to design our analysis, interpret our results, and communicate in ways that are most useful. So the way we're going to do this is work with our partners from state and tribal monitoring programs to assemble the data they've been collecting over the past several decades on fish tissue mercury. We're also going to look at factors that influence how much mercury is in systems and how available it is to fish in order to quantify that effect of zebra mussels while accounting for other factors. And so we're going to build a statistical model to understand how zebra mussel invasion might interact with other lake and watershed level characteristics to predict and understand which fish species might be elevated in mercury. We're also going to look at how those factors, including zebra mussels, might affect mercury concentrations in multiple fish species. So in the current monitoring programs, sometimes we only have the data to say, okay, mercury is elevated in one species of fish, say black crappie. Well, what can that tell us about mercury levels in other species of fish that maybe we haven't sampled? Are they tracking together? Are they different? Do zebra mussels impact one species more than others? Questions like that. And then ultimately, if we're able to quantify those relationships between lake factors and zebra mussels and fish tissue mercury, we want to use that to predict locations and species where we might expect mercury concentrations to be high and where zebra mussels are likely to have the largest effect on fish tissue mercury. And we plan to communicate these results using a variety of tools, including things like mappers. You might be familiar with the AIS Explorer that a lot of MESARC researchers have worked on. So similarly, we can produce, so this tool shows risk of invasion of lakes throughout the state. We might want to build a similar tool showing risk of high mercury exposure or identify places where we might want to look more closely at fish tissue mercury. And our partners from Glyphwick have a phenomenal mercury monitoring and communication program already. So we're going to work with them to incorporate our results into their existing communication tools to communicate risk of mercury exposure. I think we have time for questions.
This question came pretty early in the chat because I know in the beginning you were talking about what lakes were managed for walleye. Do you have an idea of the total number or what percentage out of our almost 12,000 lakes are managed for walleye?
Oops, yep, I sure do. I think it's about 1,400. I have it on my slide. Oops, there it is. So yeah, 1,435. And that number is based on DNR records of kind of where are we focusing on walleye as a managed species. So it's about 1,400.
Awesome. So what other factors determine mercury concentrations in fish?
Yeah, so many. I've learned so much because this is sort of a new area of research for me. So yes, I am always learning. So other factors, as I talked about here, how much is kind of coming in from the atmosphere and the watershed. And really interestingly, things like the amount of wetlands in the watershed really influences mercury processing and how much is available to fish. Things like sulfate, so water chemistry, things like water clarity. So mercury can be degraded by UV light. So water clarity influences how much is out there. Dissolved oxygen, both in the water column and in the sediments, really influences how much is out there. And then just kind of what the fish are eating and the mercury levels in those prey items is also a key component. So there's like a lot of complicated pathways that influence fish mercury.
So kind of thinking about mercury, we have another question. Are you going to monitor mercury in the zebra mussels as well? Or is the idea is that zebra mussels are creating these conditions that increase mercury?
Yeah, that's a great question. In the current project, we are not going to monitor mercury concentrations in the zebra mussels themselves. And yeah, the second part of the question kind of nailed it. We don't necessarily think that the pathway through which zebra mussels might be influencing mercury is that, you know, they take up a bunch of it and then the fish eat them, although that is a potential pathway. We are more focusing on how they change, zebra mussels change the lake environment in a way that just makes the mercury more available to fish, even if they're not eating the zebra mussels at all.
Okay, we have another question and 1 more minute.
Okay.
Okay, so recent work from Mille Lacs showed that water quality impacts from zebra mussels were offset by co-infestations with spiny water flea. Do you intend to look for similar offsets in mercury levels?
Yes, so we are very interested in those kinds of interactions, whether it's between multiple invaders, as was the case in Mille Lacs, or, you know, the effect of zebra mussels might depend on how many wetlands are in the watershed, or, you know, watershed area relative to lake size, things like that. So certainly, yes, we're going to use our current understanding of what influences mercury processing and availability in lakes to better understand where zebra mussels might have a bigger effect or a smaller effect. And spiny water flea will be one of those factors.
Great. Yep. So for the last lightning talk of the session, I'm excited to share an overview of our new project. And as Maddie mentioned, it's titled Surveillance and Control of Chytrid Fungi in Minnesota Amphibians. I'm Amy Kinsley. I'm an assistant professor and aquatic epidemiologist in the Veterinary Population Medicine Department in the College of Veterinary Medicine here at the University of Minnesota. And this new project is a team effort involving Jennifer Lamb, who's an associate professor from St. Cloud State University, Daniel Greer and Megan Winsler from the United States Geological Survey, Molly Bletz, an assistant professor at Penn State University, and in collaboration with Minnesota DNR's herpetologist, Carol Hall, and non-game wildlife specialists, Andrew Herberg and Krista Larson. So the massive loss of amphibians that has been recorded over the last 3 decades is known to be one of the most critical threats to global biosecurity or biodiversity. The decline has been caused in large part by the pathogenic chytrid fungus, Batrachochytrium dendrobatitis, also known as BD. So conservative reports estimate that BD has contributed to the decline of at least 501 amphibian species, making it among the most destructive invasive species. And so this figure here is showing the global distribution of chytrid mycosis-associated amphibian species declines, and that's grouped by continental area and then classified by decline severity. And so this shows just how widespread and significant this issue is. Since its detection in the late 1990s, BD has been detected in over 70 countries, including all continents where competent hosts exist. But recently, a new species, Batrachochytrium salamandivorians, has been known as BSAL, has been identified in fire salamanders, and that has led to the collapse of an entire population of salamanders in the Netherlands, and also mass mortality events throughout Europe. BSAL is a cutaneous or a skin infection, and the infection in susceptible animals leads to skin ulcerations, as you see here in these images. Infected animals may also develop lethargy or inappetence and death that occurs primarily through septicemia. But not all infected or exposed animals become symptomatic, and host responses vary. And so they're largely categorized as resistance. So that's hosts that there's no infection detectable and no disease. They might be considered tolerant, where the infection occurs, but there's no signs of disease that developed. They're susceptible, so the infection results in clinical disease and maybe some possible recovery in that animal, or lethal, where the animal becomes infected and then that results in death. And these different categories and the way different animals respond can make detection and management quite challenging. So recent efforts to assess the risk of BSAL in the United States has emphasized the need for iterative approaches. And so as conditions change and new information becomes available, those need to be incorporated in our understanding of risk. The most recent study to estimate the risk of BSAL across counties in the United States updated previous efforts by including information about captive animal imports, surveillance efforts, and new knowledge regarding captive species susceptibility information. And then that model was used to evaluate the change in risk due to regulatory action and increased knowledge of the system structure, such as import regulations, new knowledge of carrier species, and surveillance efforts again. So the study showed that the highest risk of BSAL exists along the California coast and portions of the Northeast along the Southern Appalachian region, but that combining the impact of import regulations, new knowledge about species that are carriers, and large-scale surveillance decreased that estimated risk in areas that were previously identified as high risk. And that readjusted our understanding of the spatial distribution of risk. And while intensive nationwide surveys that have been conducted in the past from 2014 to 2017 were incorporated in that study, they didn't identify BSAL in wild amphibians. But we know all too well that continuous surveillance is critical to detecting invasive species spread and outbreaks early before control and management becomes impossible. And so SNAPS is a student network for amphibian pathogen surveillance, and it's been playing an important role on that front since 2020. So SNAPS is a student network that's comprised of students, scientists, and educators, and it spans across 52 institutions. It embeds BSAL surveillance into undergraduate curricula and provides opportunities for students to engage in experiential learning while contributing to much needed surveillance. And so my co-PI and collaborator, Jennifer Lamb, joined SNAPS a couple of years ago, and I joined more recently. And to build off these efforts and position Minnesota as a leader in protecting biodiversity against Chytrid fungi, our objectives are as follows. So we intend for this project to generate a local scale risk model for Chytrid fungi across Minnesota, develop and implement surveillance networks that connect wildlife managers through the DNR and Minnesota colleges and universities. We hope to cultivate a more substantial knowledge base regarding Chytrid fungi and their potential impacts on the state that will be used in education and outreach. And so in the first objective, we aim to update and refine the current models that I described similarly to those that were the national scale models, but we want to include more specific and useful information regarding potential consequences and impacts on amphibian populations here in Minnesota. We want to do this by engaging with stakeholders such as the DNR and update the susceptible species list and add more granularity regarding amphibian sales. In our second activity, we aim to address some of the challenges of traditional proactive surveillance while providing unique educational experiences for undergraduate students. So in this objective, we propose to expand the SNAPS efforts to build out a Minnesota focus network by reaching out to colleges and university instructors that might incorporate surveillance and teaching about amphibian ecology or conservation biology into their courses. That may also include folks that focus on wildlife disease ecology and invasive species as well. We aim to enroll up to 20 instructors from Minnesota into the SNAPS program and emphasize equity and inclusion by intentionally reaching out to our tribal colleges in the state. And our third objective will contribute to the knowledge base regarding Chytrid fungi and their impacts on the state by developing educational materials for undergrads and also more broadly for citizens of Minnesota. We want to create at least one teaching module about invasive species that can be introduced to Minnesota colleges and universities and also the SNAPS network, and at least one course-based undergraduate research experience module, also known as a CURES. And so if you're not familiar with what a CURE is, they're modules that can be embedded within a course, and they enable the whole class of students to participate in the research process. They form questions and hypotheses and participate in the data collection and analysis as well. And so these experiences can help students really hone in and refine their critical thinking skills, and it increases equity and inclusion in science for underrepresented populations as well. So we anticipate there will be many positive impacts of the project. We're hoping that there's instrumental impact and that the research outputs can be used to inform response plans in the state. where we hope that there will be conceptual impacts with increased awareness among citizens of Minnesota about the risk of chytrid fungi and B-cell specifically. We hope that there's capacity building within the training and skill building for undergraduate students at universities across Minnesota and also through SNAPs in the US by sharing that information. We're hoping for enduring connectivity where researchers across the SNAPs network and between researchers in the Minnesota DNR with folks at MASERC, that continuity continues for years to come. And we look forward to keeping you in the loop on our research updates as this project progresses. So if you have any questions or are interested in participating in SNAPs, feel free to reach out to me. I had my email address here, but it looks like it's not showing up when I share screen strangely, but it is carr0603@umn.edu, or if you type in my name and UMN or MASERC, my contact information will come up. So with that, I will take any questions.
We do have a couple questions rolling into the chat. So is it possible for an amateur herper to recognize a chytrid infection?
Yeah, so I think one of the points that I brought out in the presentation really is that there's, depending upon the species, there might be variable outcomes, right? So we might have a species that is infected and can be infectious to other animals and not be able to tell just by visually inspecting the animal that it's infected. So it's important that we go out and do sampling where we're collecting the animals, swabbing the animals, and sending those swabs into the lab to get tested by PCR. And I just want to touch on the point, an amateur herper can be trained through the SNAPs program to go out and conduct sampling. All of the materials that are needed to be successful at this are provided through the program. And so me as like somebody who's newly participating in the SNAPs program have found it extremely supportive.
You kind of started mentioning this just now about how difficult it is to ID the B-cell visually. And, you know, is that where all the samples need to be sent for analysis?
Yeah, I mean, I think the main finding in an animal that is symptomatic would be those symptoms I had described earlier, right? So it could be something like lethargy or inappetence, or maybe you find dead salamanders, but even those aren't specific to chytrid infection or specific to B-cell. And so in those cases, we would still need to follow up and submitted sample. Again, even if we see lesions on an animal, so skin lesions or ulcerations on an animal, those can be caused by other things like abrasions. And so, yeah, it's best that the animals are sampled, that they're collected and swabbed, and those samples are submitted.
So thinking about this amateur herper, how to identify, you know, what is the ways that organizations can get involved if they're interested in helping out?
Yeah, so at this point, we're asking folks in Minnesota to reach out to me so I can collect your information and start developing a list. And then as this project evolves, we'll be reaching out to folks and letting them know ways that they could connect through SNAPs and also participate in receiving or integrating the educational materials into any of their courses. So at this point, I would, again, reach out to me, carr0603@umn.edu. And, or if you have any ideas about how we could reach out to other folks to get them involved, we would be open to accepting those ideas as well.
And thank you, David, Gretchen, everyone for their lightning talks today. It was really fun to hear about some upcoming research and projects that are happening here at MASERC.